Okay, let's dive in. Imagine you've got this super complex instruction manual for building something incredible, but the instructions, they're in a secret code. And the thing it builds does, well, everything. Today, we're really unpacking that. The journey from your DNA code to the proteins that make you you. We're calling this Decoding Life's Blueprint, and it's all based on a key chapter in Karp cell and molecular biology, 8th edition. Our mission is simple. Pull out the absolute core concepts from this text, giving you a shortcut to understanding how life works at its most fundamental level. And yeah, we'll even tackle that classic chicken and egg problem about life's origins.
Exactly. We're tracing the path of the central dogma, the flow of information. DNA to RNA, RNA to protein. Sounds straightforward maybe, but the cellular machinery. It's breathtakingly complex, really elegant. We'll look at what happens, but also how we figured it out, the experiments, the discoveries. And we'll connect it all to the real world, things like diseases and some revolutionary new tech.
Okay, so let's start with that big evolutionary puzzle you mentioned. DNA holds the plans for proteins, but you need proteins to copy DNA and make RNA. And you need RNA to make proteins. It feels like a loop, right? How could life possibly get started with everything depending on everything else?
Yeah, that mutual dependence was a huge hurdle for understanding the origin of life, a real paradox. But as we'll see, RNA itself seems to hold a key. There's this idea, the RNA world hypothesis. It suggests that early life might have used RNA for both storing genetic info and acting as catalysts, like enzymes. So RNA was doing a kind of double duty before DNA and protein specialized later on.
That's a fascinating solution. But way before that idea, scientists were just trying to link genes to basic traits, like Archibald Garrod back in 1908. What was his contribution?
Right, Garrod was a physician. He studied what he called inborn errors of metabolism. These were diseases you see caused by missing enzymes, like alkaptonuria, where urine turns dark. He realized people with this condition lacked a specific enzyme needed to break down a certain acid, so he made the connection. A faulty gene leads to a missing enzyme, which causes a specific metabolic problem. Groundbreaking stuff, really. But his ideas were mostly ignored for decades, a bit ahead of his time.
So the core idea was one gene, one enzyme, but it took a while for it to stick.
Yeah, it wasn't really picked up again until the 1940s. That's when George Beadle and Edward Tatum did their famous work with neurospora. That's a type of bread mold. They deliberately mutated its DNA using radiation. Then they looked for mold strains that couldn't grow on simple food unless you added a specific nutrient, like, say, a vitamin. If adding vitamin B6 let it grow, they knew the mutation had broken the enzyme needed to make B6.
Ah, clever. So that solidified the one gene, one enzyme idea, which then got refined a bit, right? To one gene, one polypeptide.
Exactly, because we learned many enzymes are actually built from multiple protein chains, or polypeptides. And each of those chains is typically encoded by its own separate gene. But the fundamental concept held. Genes carry the specific instructions for building proteins.
Okay, so that brings us to gene expression. What does that term actually mean?
Gene expression is just the whole process, really. Taking the information that's sort of sitting there passively in a DNA segment and turning it into a functional product like an enzyme or another structural protein. And the very first essential step in that process is transcription.
Transcription, and that's making an RNA molecule from the DNA template.
Precisely. And for making proteins, there's a key intermediate molecule discovered in 61, messenger RNA, or mRNA. The mRNA is basically a working copy of the gene's code. It carries the instructions for assembling the protein. But in our cells, eukaryotic cells, these first mRNA copies, the precursors, need a lot of work done on them. They need processing.
Processing, okay. And you mentioned other RNAs too. It's not just mRNA.
Oh, far from it. Cells make lots of RNA types. There's ribosomal RNA, rRNA, which is a core component of ribosomes at the protein factories, and transfer RNA tRNA, which actually delivers the amino acids to the ribosome. Plus, especially in eukaryotes, there's a whole zoo of other small RNAs discovered more recently. Many have regulatory roles.
Like controlling which genes get turned on or off.
Exactly. Things like snRNAs, sNORNAs, sRNAs, miRNAs, pRNAs. The list keeps growing. It shows just how much complex regulation is happening.
Here.
Much of it orchestrated by RNA. We're still figuring a lot of it out.
Okay, let's zoom back in on transcription itself. Making that first RNA copy. What's the main enzyme doing the work?
That's RNA polymerase. It binds to the DNA, moves along one strand, the template strand, unwinding the double helix as it goes. And while it moves, it builds a complementary RNA strand, adding nucleotides one by one. It's surprisingly fast, maybe 20 to 50 nucleotides per second, and often many polymerases transcribe the same gene at once.
Wow, like a little molecular assembly line. Is it the same process in bacteria versus, say, human cells?
Similar core idea, but different details. Bacteria have a simpler setup. Their RNA polymerase, usually with a helper called a sigma factor, binds directly to specific start signals on the DNA called promoters. In eukaryotes, like us, it's more involved. We have several types of RNA polymerase. So the one making mRNA precursors is RNA polymerase II. And it doesn't bind alone. It needs a whole team of helper proteins called general transcription factors, or GTFs. They all assemble together at the promoter region on the DNA to form what's called a pre-initiation complex.
And I've heard of the Tata box. Is that part of the promoter?
Often, yes. It's a specific DNA sequence, TATA-ish, found in many eukaryotic promoters. A key protein, Tato binding protein, or TBP, recognizes and binds to it. And interestingly, it bends the DNA quite sharply. That bending seems to help recruit the rest of the transcription machinery.
So once RNA polymerase gets going, what happens?
As it starts making the RNA chain, a part of the polymerase itself, its C-terminal domain, or CTD, gets modified. Specifically, it gets phosphorylated. This phosphorylation acts like a switch. It helps the polymerase detach from those initial GTFs, and it also serves as a docking site to recruit other proteins needed for the next steps processing the new RNA molecule, like capping and splicing factors.
Right, processing. You mentioned ribosomes are mostly RNA. How are those RNAs made?
Ribosomal RNA is incredibly abundant, over 80% of all RNA in the cell. The genes for RNA, called rDNA, are usually present in hundreds of copies, all clustered together in a part of the nucleus called the nucleolus. Think of the nucleolus as the cell's ribosome factory. The RNA starts as one long precursor molecule, like the 45S pre-RNA in humans. Then this large precursor gets precisely cut and processed into the final, mature RNAs, the 28S, 18S, and 5.8S molecules.
How does the cell cut it so precisely?
That's where another class of small RNAs comes in, the small nucleolar RNAs, or sNORNAs. These sNORNAs team up with proteins to form sNORNPs. They use base pairing to guide enzymes to the exact spots on the pre-RNA, either for cutting or for making chemical modifications like adding methyl groups or converting bases. It's incredibly specific.
Wow. RNA guiding RNA processing. What about the tRNAs, the transfer RNAs?
They have a similar story, transcribed as larger precursors, then trimmed at both ends. Sometimes an internal piece is removed, and they also get heavily chemically modified. An enzyme involved, ribonuclease of P, is fascinating. Its catalytic power, the ability to cut the RNA precursor, actually comes from its RNA component, not the protein part, another ribozyme.
Okay, this is maybe one of the biggest surprises in the whole story. For ages, everyone thought genes were just continuous stretches of code, right?
That was the assumption, yes. A gene sequence in DNA directly matched the mRNA sequence. Seemed logical. But then in 1977, experiments, particularly with adenovirus, blew that idea apart. Researchers like Sharp, Roberts, Chow, and others found that the final viral mRNAs had sequences cobbled together from different separated parts of the viral DNA.
So the DNA had extra bits in between the coding parts.
Exactly. The coding segments, which they call exons, were interrupted by these non-coding intervening sequences, now called introns. And this wasn't just viruses. They quickly found this split gene structure was common in our own genes, too.
How do they even prove that? See these introns?
One key technique was DNA-RNA hybridization. If you took the DNA gene containing introns and mixed it with its final mature mRNA, the exons of the DNA would pair up with the mRNA. But the intron sequences in the DNA had nothing to pair with, so they'd loop out. You could see these loops under an electron microscope.
Visual proof. So this means after transcription, the initial pre-mRNA needs major editing. What happens?
Three main things for eukaryotic mRNA. First, a special cap is added to the 5-foot end. Second, a long tail of adenine bases, the poly A tail, is added to the 3-foot end. And third, all those introns have to be precisely removed and the exons stitched together. That's RNA splicing. And remarkably, a lot of this capping, splicing, and tailing happens while the RNA is still being made co-transcriptionally.
Let's break those down. The 5-foot cap, what is it and what does it do?
It's a modified guanosine nucleotide added backwards, basically, in an unusual 5 to 5-footed linkage. It acts like a protective helmet, protects the mRNA from being chewed up by enzymes. It helps get the mRNA out of the nucleus, and it's absolutely critical for starting protein synthesis later on.
And the poly A tail at the other end.
That's a string of typically around 250 adenine nucleotides added after transcription finishes. It's not coded in the DNA. It also helps protect the mRNA from degradation and plays roles in translation efficiency and regulation.
Okay, now the big one. Splicing, removing those introns. You said it has to be precise.
Absolutely critical. If the cuts are off by even one nucleotide at the exon-intron boundary, it shifts the entire reading frame for the rest of the protein sequence. The resulting protein will be complete gibberish, totally non-functional. It's estimated something like 15% of inherited human diseases are actually caused by mutations that mess up pre-mRNA splicing.
That's significant. How does the cell know exactly where to cut?
There are short consensus sequences in the RNA at the 5 and 3' ends of the intron, the splice sites, usually a GU at the 5' end and an AG at the 3' end. But those signals alone aren't always enough. Other sequences, often within the exons themselves, called enhancers, help guide the machinery.
And you mentioned some introns can even splice themselves out.
Yes, group Y and group 2 introns. They can fold up into complex structures that catalyze their own removal, often forming a loop structure called a lariat. This was another huge piece of evidence that RNA could be catalytic.
But most splicing in our cells needs help, right? What's involved?
Right. Most pre-mRNA splicing relies on a large molecular machine called the spliceosome. This isn't a pre-built machine. It actually assembles step-by-step onto the pre-mRNA molecule. Its core components are those small nuclear RNAs, the snRNAs we mentioned earlier, combined with proteins. These complexes are called snRNPs, pronounced SNRPs.
SNRPs. Okay. How do they work?
It's a dynamic process. Different snRNPs recognize the splice sites and other key points on the intron. Like U1 snRNP binds the 5' splice site, U2 binds an internal branch point, then others like U4, U5, U6 join in. There are lots of RNA interactions between the snRNAs and the pre-mRNA, and also between the snRNAs themselves. It requires energy, ATP, mainly to power RNA helicases that rearrange the complex. And interestingly, it's thought that one of the snRNAs, U6, might actually be the catalytic core, another ribozyme performing the cutting and pasting reactions.
Amazing. RNA doing chemistry, RNA guiding RNA. Let's switch gears slightly to another RNA phenomenon, RNA interference or RNAi. This started with purple petunias losing their color. That sounds weird.
It was very puzzling back in 1990. Scientists added extra copies of a gene for purple pigment, expecting darker flowers. Instead, many flowers turned white or patchy. It turned out the extra gene copies somehow caused both the added gene's mRNA and the plant's own pigment mRNA to be destroyed. They called it post-transcriptional gene silencing. But they didn't know how it worked.
Until the worm experiments.
Exactly. Andrew Fire and Craig Mello in 1998, working with the nematode worm C. elegans, they injected double-stranded RNA, dsRNA, corresponding to a specific gene, and bingo, production of the protein from that gene shut down very effectively. Single-stranded RNA didn't work nearly as well. They coined the term RNA interference.
So double-stranded RNA is the trigger. Why would cells react to that?
The thinking now is that dsRNA is often a sign of something foreign or unwanted, like a replicating virus or active transposons, those jumping genes. So RNAi likely evolved as a kind of genetic immune system to recognize and destroy these potentially harmful nucleic acids.
How does the destruction happen? What's the mechanism?
It's elegant. First, an enzyme called dicer signs the dsRNA and chops it into small pieces, about 21-23 nucleotides long. These are called small interfering RNAs, or siRNAs. Then, one strand of the siRNA gets loaded into a protein complex called RISC, the RNA-induced silencing complex. The siRNA acts as a guide. It directs RISC to find messenger RNAs in the cell that have a matching sequence. Once it finds a match, an enzyme within RISC, usually an argonaute protein, cuts the target mRNA, leading to its degradation. Poof, no protein made from that message.
Okay, the potential for using this therapeutically seems huge if you want to shut down a problematic gene.
Exactly. Instead of designing a drug to block the protein, you could design an siRNA to just destroy the mRNA before the protein even gets made. A major advantage is specificity. You can design siRNAs to match only one specific gene's mRNA, potentially reducing the off-target side effects you often get with traditional drugs.
So it's become a big research tool.
Massive. It's a standard way now to knock down a gene's activity in the lab and see what happens. Helps figure out what genes do. And clinically, yes, there's huge investment. Trying to develop siRNA drugs for cancer, viral infections, genetic disorders.
What are the main roadblocks for getting these drugs to work in people?
Delivery is a big one. Getting these RNA molecules into the right cells in the body and keeping them stable long enough is tricky. It's easier for localized treatments, like in the eye or maybe inhaled into the lungs. For systemic diseases, researchers are working on nanoparticle delivery systems, sort of like tiny envelopes to protect and target the siRNAs. There have been clinical trials, some successes, some setbacks. It's still an active area, but the promise is definitely there.
Now, siRNAs aren't the only small regulatory RNAs, are they? You mentioned miRNAs.
Right, microRNAs. Their discovery also started in C. elegans back in 1993. Scientists found a tiny RNA encoded by a gene called Lin4 was vital for normal larval development. And it worked by binding to the mRNA of another gene, not causing it to be cut, but just blocking it from being translated into protein.
So silencing, but in a different way.
Exactly. Then another one, latnasomdetting, was found, and it turned out to be conserved across many species, including humans. That really opened the floodgates. Myrnas are similar in size to sirenase, made by similar machinery involving dicer, so they're often called cousins.
But they have different origins and jobs.
Generally, yes. Sirenase often come from external sources like viruses acting as defense. Myrnas are encoded in our own genome. They're part of our normal gene regulation toolkit, fine-tuning the expression of potentially thousands of our own genes. They play roles in development, cell growth, metabolism, you name it.
How are they made?
They start as longer primary transcripts made by RNA polymerase II, which fold into hairpin shapes. An enzyme called drosha processes them in the nucleus, then they're exported, and dicer does the final cut in the cytoplasm to make the mature, double-stranded myrna. Like sirenase, one strand gets loaded into a RISC complex.
But you said they often block translation instead of cutting?
In animals, yes. Myrnae often bind imperfectly to their target mRNAs, usually in a region called the 3' untranslated region. This partial mash typically leads to the ribosome stalling, or the mRNA eventually being degraded, rather than immediate cleavage. It's estimated humans might have over 1,000 myrnas, regulating maybe a third of all our genes. It's a huge layer of control.
Okay, and quickly, what about purnas?
PUE interacting RNAs. These are a bit longer, found mainly in germ cells, sperm and egg precursor cells. Their main known job is to fight transposons, those jumping genes. They silence them to protect the integrity of the genome that gets passed to the next generation. Their production pathway is different too. It doesn't involve dicer.
Got it. And then there's CRISPR. Everyone's heard of CRISPR now, but it started as a bacterial defense system too.
That's right. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It's basically a section of the bacterial genome where the bacterium stores little snippets of DNA from viruses that have previously attacked it, like a most wanted gallery. These viral snippets are separated by repeated sequences.
A bacterial immune memory system. How does it use that memory?
When a virus whose sequence is stored in the CRISPR array attacks again, the CRISPR region is transcribed into RNA. This RNA is processed into short guide RNAs, ECAR RNAs. Each guide RNA combines with a CRISPR-associated enzyme, the most famous being Cas9. The guide RNA then directs the Cas9 enzyme to the invading viral DNA that matches the stored snippet. And Cas9 acts like molecular scissors, cutting the viral DNA, neutralizing the threat.
And the huge breakthrough was realizing we could hijack this system.
Precisely. Scientists figured out you could design your own guide RNA to match almost any DNA sequence you want. Then, if you introduce that guide RNA along with the Cas9 enzyme into a cell, bacterial, plant, animal, human, Cas9 will be directed to cut the cell's DNA exactly where the guide RNA matches.
And cutting the DNA lets you edit the genome.
Yes. The cell tries to repair the cut. Sometimes it makes errors, introducing small mutations that can disable gene. That's gene knockout. Or you can provide a template DNA sequence, and the cell might use that to repair the break, effectively inserting or correcting a gene sequence. That's gene editing. It's made genome modification much easier, faster, and more precise than older methods. It's revolutionized genetics research and has huge therapeutic potential.
All these different RNAs, siRNAs, mRNAs, purnas, CRISPR RNAs, it really changes the picture of the genome, doesn't it? It's not just about protein coding genes.
Absolutely. The discovery of all these non-coding RNAs, plus findings that vast stretches of the genome were transcribed, even if not into protein, led to the concept of the transcriptome being much bigger and more complex than we thought. We now know about long non-coding RNAs, LNC-RNAs too, hundreds or thousands of bases long, that regulate gene expression in various ways, often by interacting with chromatin. There's still debate about how much of this pervasive transcription is functional versus just noise, but it's clear RNA plays many more roles than just being a messenger. We're still discovering them.
Okay, let's circle back to the central process, making proteins. Before translation happens, we need to understand the code itself, the genetic code. George Gamow figured out it had to be triplets.
Yeah, back in the 50s. Simple logic, really. You have 4 RNA bases, A, U, G, C, and 20 amino acids to specify. If the code word's codons were only 1 base long, you'd only have 4 codes. Not enough. If they were 2 bases long, 4 times 4 gives 16 codes. Still not enough for 20 amino acids plus start-stop signals. But if codons are 3 bases long, 4 times 4 times 4 gives 64 possible codes. Plenty. So the code had to be read in groups of 3 nucleotides. Crick and Brenner later confirmed this experimentally.
And when you look at the genetic code chart, you see multiple codons often code for the same amino acid. That's degeneracy.
Right. Most amino acids are specified by more than one codon. For example, leucine has 6 different codons. This degeneracy provides some buffer against mutations. A change to the third base of a codon often doesn't change the amino acid specified.
And the molecules that actually read the code are the transfer RNAs, the tRNAs.
Exactly. They're the adapters, the decoders. They physically link the codons on the mRNA to the amino acids they specify. Each tRNA molecule has 2 key features: an anticodon loop with 3 bases that are complementary to an mRNA codon, and an attachment site at the other end where its specific amino acid is loaded on.
How does the correct amino acid get attached to the correct tRNA? That seems crucial for accuracy.
Extremely crucial. That job is done by a set of enzymes called aminoacyl tRNA synthetases. There are typically 20 different synthetases, one for each type of amino acid. Each synthetase enzyme is highly specific. It recognizes its particular amino acid and all the corresponding tRNA molecules, remember degeneracy, that should carry that amino acid. It chemically attaches the amino acid to the tRNA. This is called tRNA charging.
Okay, so we have the mRNA message, we have the charged tRNAs ready with amino acids. Now finally, the ribosome, the protein factory.
Right. The ribosome is where it all comes together. It's a complex machine made of ribosomal RNA, rRNA, and proteins. It has binding sites for the mRNA and for the tRNAs. Typically described as 3 main sites: the A amino acyl site, the P peptidyl site, and the E exit site.
How does protein synthesis actually start? Initiation.
It begins with a small ribosomal subunit binding to the mRNA near the start codon, which is usually AUG. Then a special initiator tRNA carrying methionine or formylmethionine in bacteria binds to that start codon in the P site. Finally, the large ribosomal subunit joins the complex. This whole process requires helper proteins called initiation factors.
And eukaryotic initiation is a bit more complex again?
Yes, it involves more initiation factors, and it recognizes the 5-foot cap on the mRNA to start scanning for the correct AUG start codon.
Okay, the ribosome is assembled, the first tRNA is in place. Now elongation, adding amino acids one by one. How does that cycle work?
It's a 3-step cycle. First, a new charged tRNA carrying the next amino acid specified by the codon in the A site enters that A site. This requires elongation factors and energy, GTP. Second, the crucial step: peptide bond formation. The ribosome catalyzes the formation of a bond between the amino acid on the tRNA in the A site and the growing polypeptide chain attached to the tRNA in the P site. This transfers the growing chain onto the tRNA in the A site.
And this is where the ribozyme activity comes in.
And this is it. For decades, it was assumed a ribosomal protein catalyzed this peptide bond formation. But structural studies, especially X-ray crystallography, revealed something stunning. The active site where the chemistry happens is composed almost entirely of ribosomal RNA. There are no protein side chains close enough to directly participate in catalysis. The RNA positions the tRNAs perfectly for the reaction to occur, likely involving a specific part of the P site tRNA itself. So the ribosome is fundamentally a ribozyme. Its core catalytic function is performed by RNA.
Mind-blowing. RNA building proteins. What's the third step after the peptide bond forms?
Translocation. The ribosome moves exactly 3 nucleotides, 1 codon along the mRNA in the 5 to 3 minute direction. This movement shifts the tRNAs. The now empty tRNA that was in the P site moves to the E exit site and leaves. The tRNA holding the growing polypeptide chain, which was in the A site, moves into the P site. This leaves the A site empty again, ready for the next charged tRNA to come in. This movement also requires an elongation factor and energy.
Then this cycle repeats, adding amino acids until
the ribosome encounters one of 3 specific stop codons in the mRNA: UAA, UAG, or UGA. There are no tRNAs with anticodons matching these stop codons. Instead, when a stop codon lands in the A site, proteins called release factors bind there. These release factors trigger the ribosome to cut the completed polypeptide chain free from the last tRNA. Everything that disassembles the ribosomal subunits, the mRNA, the release factor is ready to start again.
You mentioned quality control earlier, like NMD.
Right, nonsense-mediated mRNA decay. It's a surveillance system that detects and destroys mRNAs that have a stop codon appearing too early, a premature stop codon. This is often beneficial, preventing the cell from making truncated, possibly toxic proteins from eucated genes. But sometimes, like in certain genetic diseases, cystic fibrosis, muscular dystrophy, NMD can be harmful because it destroys an mRNA that might have otherwise made a shortened but still partially functional protein. So there's research into drugs that could interfere with NMD in those specific cases.
And cells can make proteins faster by using polysomes.
Yes, polyribosomes or polysomes, that's just when multiple ribosomes translate the same mRNA molecule simultaneously. You get a whole string of ribosomes moving along the mRNA, each one making a copy of the protein. Greatly increases the output.
So the discovery that the ribosome is a ribozyme really solidified the RNA world idea, didn't it?
It was incredibly powerful evidence. Those structural studies by Jonath, Stites, Ramakrishnan, and others were monumental. Seeing the atomic details, realizing RNA formed the catalytic core for life's most central process, protein synthesis, was a huge boost for the hypothesis. It showed RNA can catalyze complex reactions, making it plausible that it could have supported early life before proteins became the dominant catalysts.
And DNA became the main information store?
Exactly. Over evolutionary time, you got specialization. DNA for stable information storage, proteins for versatile catalysis, and RNA bridging the gap, but also retaining many crucial catalytic and regulatory roles. And we can even create new ribozymes in the lab now through directed evolution, showing the catalytic potential of RNA is vast.
Wow, what an amazing, intricate story we've unpacked today. From wondering how life even started, to seeing the ribosome's RNA core at work, to editing genomes with CRISPR, it really gives you a deep appreciation for how that information in DNA gets expressed through the incredibly versatile molecule that is RNA.
It really does. We've seen RNA as a messenger, a decoder, a structural component, a regulator, a defense molecule, and a catalyst. The complexity is stunning, and discovering new types of non-coding RNAs just keeps reminding us there's probably still more hidden complexity within our genomes.
It definitely makes you think. What other roles is RNA playing right now that we haven't even discovered yet? And how will understanding these fundamental processes continue to drive new technologies in medicine and biotechnology? We hope this deep dive into Karp's chapter gave you a solid foundation and maybe sparked some curiosity. Thanks for joining us today as we decoded life's blueprint.